Robert Hooke was the first person who looked at cork under a microscope. He decided that the boxes that made it up looked like cells, giving the cell the name cell. Rudolf Virchow said cells come from other living cells. He thought every cell came from an ancient cell. A cell that splits is almost identical to the other. Matthew Schleiden found out that as well as animal cells, plants were made of cells too. Theodor Schwann discovered that animals are made of cells and discovered how the neuron works. Anton van Leeuwenhoek first looked at living organisms under a microscope. Janet Plough found out that the structure of the cell membrane. Every cell has a membrane and cytoplasm. The membrane can be penetrated, opened by protein gates, or can be opened temporarily for bigger things. All of these ideas added up to be the cell theory. All living things are composed of cells. Cells are the basic structure and function of living things, and new cells are produced from existing cells. Microscopes are used to view a subject under a slide. They allow you to zoom in and see the smaller parts of the subject. Cells were first discovered using a microscope. Different sized nose pieces allow you to magnify something more or less. The stage clips hold the slide in place, while the stage can be moved up and down using knobs. There are two major types of cells, prokaryotic and eukaryotic. The only thing they have in common is that they both have a cell membrane and cytoplasm. Prokaryotic cells are jumbled inside, smaller and simpler than eukaryotic, don't have a true nucleus, can absorb foreign DNA, contains DNA in the cytoplasm. Usually they are bacteria and algae. The bacteria are extremely different from each other. On the other hand, eukaryotic cells are compartmentalized and more organized. They're complex and have a true nucleus. They can be multicellular organisms such as plants, animals, and microorganisms. The cell can be considered like a city, surrounded by a fence like the cell membrane. In plant cells, there's extra fortification like a barbed wire fence, called the cell wall. There's a cytoskeleton inside the cell that's used as support and transport. The microtubules are hollow and can be traveled on the inside or outside of. It's called the endoplasmic reticulum. The vessels that travel along the road can contain things, such as amino acids. The amino acids can be built using the recipe or DNA that ribosomes send to it. However, if the amino acids are built without first visiting the Golgi bodies, they'll be destroyed by lysosomes. In animal cells, there are a lot of lysosomes because there's a lot of waste to get rid of. However, in plant cells, there aren't as many because the vacuoles store all the waste and water and all the nutrients the plant needs. The vacuoles in an animal cell are much smaller because as they don't need to store the waste because they have the lysosomes to get rid of it. The nucleus can be considered the brain of the cell. It stores all the DNA. Near the end of the cell's life, it needs to sort the DNA. At first, it's in a jumbled mess called chromatids. However, the DNA gets sorted into chromosomes. Each half of a chromosome is called a chromatid. The two sides are identical. A gene is in a section of DNA on a chromosome that controls a certain trait. When the cell successfully replicates all the DNA, it divides with the help of centrioles. The mitochondria is the powerhouse of the cell. Plants have mitochondria also, but they also have chloroplasts. The chloroplast can be considered like a solar center. It takes solar power and converts it into energy for the plant. Small molecules such as water can enter the cell through pores. However, larger molecules, like things other than water, need to enter through protein gates. The protein gates are shape specific, so the cell has to be the right size to be able to fit through. The cell is full of something called cytoplasm. Although slower than traveling on the endoplasmic reticulum, the cytoplasm can be traveled through using a system of currents. Osmosis is the diffusion of water through a semi-permeable membrane. Osmosis is slow, which is why a small cell size is ideal. Isotonic is when there's the same concentration inside and out of the cell. Things go into the cell and leave the cell at the same time. Hypertonic is when a less concentrated substance is inside the cell and moves to a higher concentration outside. Hypertonic diffusion is when a more concentrated solution is inside the cell and moves outside. Whenever a solution enters the cell and makes it too crowded, it bursts. This burst is called lysis. When the cell needs a higher concentrated substance inside, it requires energy to hold it that way. This is called active transport. Facilitated diffusion is when sugars and amino acids also need to enter the cell. Although water can get through the cell membrane, other macromolecules need to use the protein character modules, or a protein gate. Endocytosis is when things enter the cell. However, exocytosis is when things leave the cell. Same process of endocytosis, but backwards.